Hi, I'm a germinal epithelium cell, and today I'm going to show you how I, a sex cell, undergo meiosis. So yeah, let's go! Before I undergo meiosis, I first undergo mitosis, which involves me turning from one diploid cell into producing two diploid cells. Before meiosis, I'm also involved in the interphase, in which I will grow bigger from a germinal epithelium cell into a primary spermatocyte, as well as replicating my DNA. Meiosis starts with meiosis 1, and the first step in meiosis 1 is prophase 1. In prophase 1, chromosomes in the nuclear membrane condense, allowing the chromosomes to be more visible. Synapsis then occurs, which involves homologous chromosomes to pair side by side. During synapsis, crossing over happens to the pair of homologous chromosomes at an area called the chiasma. After crossing over, the nuclear membrane surrounding the chromosomes disintegrates, with centrioles migrating to opposite poles, and then extending to form spindle fibers. The sister chromatids then attach to the spindle fibers in prometaphase 1. Next, I'm going to go through metaphase 1. In metaphase 1, the spindle microtubules move the bivalent chromosomes to the equator of the cell, making the chromosomes align randomly on the metaphase plate due to independent assortment. In anaphase 1, the homologous chromosomes separate through the shortening of the spindle fibers. One chromosome of each pair moves to the opposite poles in the cell. Meiosis 1 ends with telophase 1, in which chromosomes uncoil and the chromosome number changes from diploid to haploid. The nuclear envelope reforms at each pole, followed by cytokinesis, which separates the cell into two daughter cells with a cleavage furrow, resulting in two secondary spermatites, which are two haploid cells. Meiosis 2 then follows meiosis 1. The first step of meiosis 2 is prophase 2, in which chromosomes, which consist of two sister chromatids, condense and become visible, while centrioles migrate to opposite poles and reforming spindle fibers and the nuclear envelope disintegrates. In prometaphase 2, the spindle fibers will attach to the centromeres of the sister chromatids, with the sister chromatids attached to opposite poles. In metaphase 2, the sister chromatids are aligned along the equator of the cell by the spindle fibers in preparation for centromeres to divide in anaphase 2. Next is anaphase 2, in which the sister chromatids separate by splitting their centromeres. The resulting sister chromatids will move to opposite poles of the cell when the spindle fibers shorten. Finally, in telophase 2, the chromatids reach the opposite poles of the cell. They decondense and a nuclear envelope forms, resulting in 4 haploid daughter nuclei. Cytokinesis occurs, dividing the 2 haploid daughter cells into 4 haploid daughter cells with a cleavage furrow. Because I was a germinal epithelium cell, for me, I would first turn from a diploid germinal epithelium cell into a diploid primary spermatocyte after interphase, and then from a diploid primary spermatocyte into 2 haploid secondary spermatocytes after meiosis 1, and then from 2 haploid secondary spermatocytes into 4 haploid cells, which will be known as spermatids. As spermatids, we will attach to a Sertoli cell to transform ourselves into the final form of our development, which is spermatozoa, through specialization. We will then finally leave the testes after we mature. So yeah, this is my experience of the meiotic process as a germinal epithelium cell. I hope this is helpful. Okay, bye!